Hello, everyone, and welcome to the talk on Turning Science into Medicine, part of the Modern Cell-Based Assays Virtual Symposium. I am Lisa from Nextland Bioscience, where we have a team of experts in the science of cell counting and cell-based assays, and I will be your moderator for today's event. Before we begin, I would like to remind everyone that this is an interactive event. We encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. I now present our first speaker, Dr. Jean Chu, Chief Technology Officer and founder at Nextland Bioscience. For a complete biography on Jean, please visit the Biography tab at the top of your screen. So without any further ado, I would like to kick things off by welcoming our speaker, Dr. Chu.
Good morning, good afternoon. Today, I'd like to talk about unique image cytometer with advanced capabilities for high-speed, versatile cell-based assays. When we do cell-based assays, we have a few options. If your cells are grown, like the image upper left, if you do the flow cytometer, you trypsinolyze your cells, and you send them through a stream of liquid where each cell becomes a dot on your result plot. Sometimes we cannot do that because biological features cannot be trypsinolyzed and sent through the liquid flow. We do this on microscope. Images are acquired. If we want to quantify them, most likely we are manually doing this under a microscope. Image cytometry means to turn cell images into numerical data. The question is, can we do that by design systems to directly take cells in a microtiter plate, image in a machine, produce cell images, apply imaging analysis software to quantify the image and come up with different features, objects, apply gating mechanism to classify them, produce numerical result. Zyligo imager is one of the such systems. It has 5 imaging channels and special optical designs. Diving a little deeper into what these designs are, it consists of a high-speed scanning goggle mirrors, large area uniform illumination light source, multiple auto-focusing methods, large focal depth for 1 micron per pixel image resolution, has the option to select scanning resolutions at a different micron per pixel level. It accommodates many different forms of tissue culture vessels. So with these designs, make it this a unique system, different from microscope design, and different from a lot of imagers automate the microscope. So what capabilities do these designs bring us? It brings us auto-stitched whole well cell images at the 1 micron per pixel. You hear me repeating the 1 micron per pixel. The significance is it allows us to quantify very small cells. So we don't just want the cell images, we want numerical data. And this is provided by simultaneously image and analyze. So when you finish imaging the 96-well plate, you also get your cell count. A lot of times, suspension cells pose a challenge for imagers because they move around when the plate is moved on the imager. So it is very important for us to be able to scan image suspension cells. So when a 3D spheroid is presented to the imagers, microscope-based design requires acquiring a stack of images, then collapse them to come out with a single image for quantification. There is no need to do it on this imager. It's just one image does the job. There are multiple fluorescent channels give us opportunity to quantify subpopulation of cells with different fluorescence or different combinations of fluorescent markers. So it is important to have a built-in gating software like we do in flow cytometry. So this is a quick summary of various different well format and speed performance for them, produced about 3 years ago. Since then, there are improvements for all of it. And it is important to note, we don't just stitch whole well image for 96 well, we also present for 24 well as well as 6 well. Next, we go into quantification using bright field images, doing it in a label-free manner. So it depends on your cell. There are different imaging analysis methodologies. On the upper left, we provide cell counting for the cells have distinct membranes. When the cell grows in clumps or colonies, we use confluence, which is a measure of areas occupied by the cells as comparing to the total well area. It is interesting to apply the high quality bright field image to document a single cell in a well grow into a colony to prove the monoclonality. So when we are doing 3D models, we not only just want to look at 3D tumor as 3D one sphere, we also want to look for the invasion or migration of cells from the spheroid. It is important to do the quantification as well. So next we go into a little bit of detail about using this label-free bright field imaging technique to quantify T cell. So it is critical to produce the whole well image to count every cell in that well, because a lot of times, especially at a low concentration, cells are unevenly distributed within the well. The middle panel shows the different concentration of cells, number of cells in the well, going from over 100,000 on the left all the way to a few cells or less than 100 cells on the right. So as I mentioned many times, we don't just want the images of cells, we want numbers. The lower panel showed you the data report from Cyligo directly in the software. You see the number of cells as well as the heat map to indicate the numerical number of cells counted. So with this, the same plate can be scanned in multiple days. The upper right upper left show you the growth curve generated for every well of a T cell proliferation assay, clearly seeing certain rows have no proliferation versus other ones has a lot more proliferation. It is also important to measure cell size, plot the size distribution for each for the cell population for each well. It is utilized for a T cell activation screening, and in the lower right corner, you see the growth curve, and you can clearly see by plotting the cell population with a larger size per well, we can identify protein X enhanced T cell activation. It is important to remind everyone to conduct this measurement, we took 5 minutes per 96 well plate. The same kind of assay can be applied to look at the immune cell chemotaxis. Cells migrate through a membrane to the bottom of the plate, easily counted. Be able to count low number give us much better assay dynamic range. Same dynamic range is required for conducting serum neutralization assay, for infectivity detection, for the vaccine, as well as the viral, antiviral development. Low number of cells in the 96 well without trypsinization gave the sensitivity to utilize this for very low infectivity measurement. So sometimes we don't look at a single cell as the assay readout. We look at larger biological features. This example is looking at the viral plaque, which is the damage zone caused by viral infection on a healthy monolayer of cells. In this experiment conducted in 24 well plate, we count the number of viral plaques, measure the area, and the other morphological features. The experiment is used to identify a vaccine candidate by systematically doing genetic modification of the viral genome. So you can imagine there are a lot of plates get screened, and imagine how quickly scanning, quantifying, doing it automatically is very significant productivity for that project. So now we switch to fluorescent assays. There are many fluorescent labels developed for many cell functions, as well as for protein expression analysis. Here I gave a couple examples. To do cell cycle analysis for both suspension and adherent cells, we can fix the cell, fix, permeabilize, and stain with propylene iodide. So the plate is then imaged. For every well, every cell, we measure the fluorescent intensity, plot the DNA content with cell population. Alternatively, EDU or BRDU is used to label the S-phase cell population. It is also important to note fluorescent proteins are increasingly used as markers for gene editing of cells. Switch gear to talk a little bit about cell-mediated killing assays. In this case, not only we want to plot the reduction of tumor cells, as indicated by the lower portion, that is instead of proliferation curve, now it's a killing curve. Not only we want to produce that curve for each well, we also want to look at immune complex formation using bright field imaging. So imager in this case can give us both the cell count as well as the other morphological features. So there is endless different kind of biological assays conducted using this imager. Some example here are fluorescent plaques, wound healing quantification, spheroid viability. This is a beautiful picture. What it really tells us is that many different fluorescent proteins are utilized as markers, as a toolkit for CRISPR-based gene editing. And in this case, a population of cells are mixed together. We use these models for co-culture experiment. Quickly summarize, imaging cytometry can be utilized, give us many different cell-based assays. On the left side, we just use bright field. On the right side, we use fluorescence, but in the middle, we combine all the different imaging techniques to produce additional assays, additional assay capabilities. Some examples here are latest applications, such as organoids for quantification for 3D cultures, immune cell phenotyping for CAR T development, as well as pure and simple super high speed throughput cell counting. We do a lot of cell counting, cell enumeration. I talked quite a bit on the pink portion of this graph. We enumerate cells in the microtiter wells. But one other important work we do for all sorts of measurement is cell counting and cell variability measurement, depicted by the method provided on the top portion, the green portion. We take sample out of various different vessels during bioprocessing, during patient sample preparation. We do the cell variability measurement and live cell concentration measurement. So latest advancement is to provide a system, do these measurement really fast and really high throughput. So here we are going to count 24 samples in 2 minutes. So with this, I'll conclude my talk.
Okay,
Next,
good. Oh, sorry.
Next, I'm going to introduce our speaker.
So hi, good afternoon, everybody. I just asked the question, can you hear me okay? Hopefully you can hear me okay. So my name is Steve Rees. I am Vice President of the Discovery Biology Group at AstraZeneca. And what I'd like to do over the next 20 minutes or so is talk to you about some of the work that we're doing at AstraZeneca to turn science into medicine, some of the challenges that we face in terms of improving the success of our discovery portfolio, the critical role that cell-based assays and advances in cell-based assay technology play in terms of our ability to increase our success in drug discovery. And I'll also talk about one area of new activity, the science of functional genomics, that we are now using broadly to identify new drug targets to bring into our portfolio. As I start the presentation, the first thing I'd like to reflect upon is we hear a lot about the challenges facing the pharmaceutical industry. We hear a lot about the ever-increasing cost of the medicines. We hear a lot about the so-called failure of the pharmaceutical industry. But I'd just like to start maybe with a couple of very positive comments. Across 2017, 2018, and 2019, no less than 150 new medicines were approved by the FDA. 150 new medicines. That is 3 of the most productive years since FDA approvals initiated in the 1940s. And certainly through 2020, we have seen dramatic activity across pharma as a consequence of the COVID pandemic to bring new vaccines and new monoclonal antibody therapeutics through to patients. And of course, we had the fantastic data yesterday from Pfizer from the initial results from the phase 3 clinical study of their messenger RNA vaccine, demonstrating quite remarkable efficacy in patients. So I think we work in an area of, I would argue, unprecedented success in terms of our ability to bring medicines to patients. But notwithstanding that, pharmaceutical discovery remains incredibly challenging and remains a science, remains an activity in which much of what we do fails. And that's really captured in this chart. So this chart, in this data, what we've done is ask the question, for those projects that have closed at AstraZeneca, what was the reason for those projects to close? And we've done this in 2 time periods, 2005 to 2010 and 2012 to 2016. And we've asked the question for projects that closed in discovery, so that's prior to candidate selection, and those projects that failed in phase 2 clinical studies. And while the data is a little old now, I don't believe the trends have changed. So if we look at the failure in the early time period, in discovery, the primary reasons for failure were chemistry. We weren't able to identify a small molecule with the appropriate chemical properties to deliver a drug candidate, followed by changes in organizational strategy and some failures for safety. And then as we moved into phase 2 clinical studies, a large number of projects, about a third of projects failed for safety, 2/3 for efficacy. And by efficacy in a phase 2 study, we mean that the medicine did not deliver the therapeutic benefit in the patient that we hypothesized it would. As we move to the latter time period, we now move to a situation where in discovery, very few projects close for any reason other than efficacy. So we're largely able to find a molecule that we can progress to the clinic. The number of closures for strategy has decreased and safety has largely been eliminated as a failure in discovery. Whilst in phase 2 clinical studies, again, the primary driver for failure is efficacy, with very few projects closing for other reasons. So this failure for efficacy in both discovery and in phase 2 clinical studies, in essence, what that translates to is that the biological hypothesis that led us to believe that that medicine would have an effect in those patients was wrong. Either that, or we selected the wrong patient population in which to conduct that phase 2 study, and hence we recorded a negative result. So to improve our success as we move forward, what is critical is that we're able to better understand disease. Sounds very obvious, we need to better understand disease, such that we can then better understand targets, drug targets, which when we intervene using a small molecule, an antibody or another therapeutic modality, are more likely to give us efficacy in the clinic. It's worth commenting that over this time period, our overall success in AstraZeneca doubled. In the early time period, we had to start 6 projects to deliver a drug candidate. In the latter time period, we had to start 3 projects to deliver a drug candidate, and we see similar increases in success in the clinic. So the strategies we're working on in AstraZeneca, what are we doing to try and increase the success of drug discovery? So the first is a fairly fundamental statement. How can we improve our understanding of disease? If we truly understood disease, if we truly understood disease processes, we would be able to select our drug targets, we would be able to select our patient populations such that we could deliver medicines that would succeed every time. The fact that we don't means that there are many diseases, and of course CNS diseases are a prime example, where we don't understand well enough the fundamental biology of those diseases. So can we improve success through the discovery of novel drug targets? And I'm going to talk about this at length in this presentation. There is a huge effort across the industry to use technologies such as genetic association of disease, functional genomics, and AI methods to identify novel drug targets that are related to specific disease or disease segments that are more likely to succeed in the clinic. If we can identify a validated drug target, the next question we ask ourselves is, can we then deliver a therapeutic modality to that target? And of course, most of the medicines in the clinic today are either small molecules or antibodies. The medicines of the 2030s will be small molecules, a whole variety of different affinity reagents, a variety of nucleotide therapeutics, from messenger RNAs to siRNAs and the like, to cell therapies, to gene therapies. And we're certainly moving to a situation where we believe at AstraZeneca that every drug target becomes tractable through deploying the appropriate therapeutic modality to allow it to advance to the clinic. Can we apply artificial intelligence to improve success and better understand disease heterogeneity? Can we use AI in chemistry to optimize our lead molecules? Can we use AI to improve clinical trial design? And the answer to all of these questions, of course, are yes. And then how can we better use patient data to inform discovery? How can we use patient data to help us design improved assays within the discovery phase, such that we're identifying targets and molecules that are more translatable to disease? And how can we use patient data to better stratify the patient population, such that we can better deliver medicines to patients that need them? So a series of huge questions. And where I work in discovery, I work in early discovery, as I guess do many people on this call. And much of what we're focused on in early discovery is how do we identify new drug targets? How do we validate those drug targets? And how do we then find small molecules or other therapeutic modalities that allow us to progress those molecules to the clinic? And cell-based assays critically underpin everything that we do. They've always underpinned everything that we've done, moving back into the 1960s, 50s, 40s, where the vast amount, well, all drug discovery was performed on tissue slices, animal slices in traditional pharmacology experiments. So we, as everybody, are deploying cell-based assays in target discovery. We employ those assays in identification and lead optimization, and we deploy those assays in translational science. And I would argue we're very much living in a period of revolution in terms of our ability to identify and characterize and develop novel cell-based assays. A statement that is absolutely true is that every scientist that has worked in drug discovery across the last probably 100 years, certainly across the time period of my career, across the last 25 years or so, has worked on cell-based assays that they believed were translatable to disease. And more often than not, we were wrong. Those assays weren't translatable to disease. So how can we create assays that are more translatable to disease, that better mimic that disease state, such that we're able to improve our success in the identification of targets, and we're able to improve our success in the identification of molecules with activity at those targets? And I describe here 4 technologies that by and large did not exist a decade or so ago that are now embedded in discovery and helping us to create improved cell-based assays. So the first of these, of course, is stem cell biology. The ability to use iPS cells, human embryonic stem cells, to create cell-based assays that better mimic adult human cells. And perhaps the biggest impact of stem cell-based assays on drug discovery today has been within the safety arena, where models of cardiotoxicity and hepatotoxicity generated using stem cells have been critical in terms of allowing us to identify and mitigate cardiotoxicity, hepatotoxicity, and other properties in molecules as they move through development. Over the last few years, we've seen massive advances in omics technologies. So obviously DNA sequencing, with the ability to now sequence a genome for just a few $100, transcriptomics, proteomics, metabolomics, and other omics methods to create this so-called science of multi-omics. And what multi-omics allows us to do is to take disease tissue from multiple patients, and to take disease tissue in a longitudinal fashion as disease progresses, and characterize that tissue at the molecular level in terms of the transcriptome, the proteome, the metabolome, to understand what disease looks like. And with that information, we can use that information to populate so-called artificial intelligence knowledge graphs to help identify new drug targets. But we can use that information to help us develop better cellular assays in discovery that better mimic disease. Of course, in 2012, we had the discovery of CRISPR from Emmanuel Charpentier, Jennifer Doudna, and others, which was very recently recognized with the Nobel Prize. And over those 8 short years, the ability to use CRISPR to create genetically engineered models of disease, genetically engineered animal models, has again revolutionized every drug project. And through the use of CRISPR, we're able to create isogenic cell lines for studies in oncology. We're able to tag proteins. We're able to introduce mutations into proteins and, of course, delete proteins. And then bringing these technologies together, we now have the ability to create so-called complex cultures, and Jean touched on some of this. So co-cultures, organoids, 3D cultures, to create structures that better mirror disease. And the vision, of course, is ultimately, is this going to work? Maybe this slide isn't going to work. Ultimately, the vision is to so-called create a human body on a chip. Could we use 3D organoid type systems, microphysiological culture systems, to create a so-called human body on a chip, where we have a tissue that represents the human liver, the human heart, the human kidney, the human brain, the human lung, with artificial blood flowing through those systems that allow us to characterize compound efficacy. Now, that may be many years away. That may still be very much an aspiration, but I suggest that aspiration will become reality sooner than we all think. But in the interim, we certainly have the ability to create improved models of disease for specific tissues that we can apply in drug discovery. So what I'd like to do for the next 3 or 4 slides is just talk a little bit more about the work that we're doing in AstraZeneca to identify and validate the right drug targets. We and others investing very heavily at the moment in terms of trying to understand disease such that we can identify drug targets, novel drug targets that sit in the traditionally undruggable target genome that may allow us to develop medicines with improved efficacy. And we're doing that with 3 technologies. Through the ability to sequence genomes for as little as $1,000, we have an aspiration in AstraZeneca to sequence the genomes of 500,000 patients on AstraZeneca clinical trials to better understand those mutations that are associated with disease, so-called rare variant analysis, such that we can identify those drug targets which, when we're able to modify, may have efficacy in those disease states. We're complementing that with investments in artificial intelligence. We have a collaboration with Benevolent AI, which allows us to mine the world's data in an agnostic fashion to identify new hypotheses that relates targets to disease. And internally and through collaboration, we're working with CRISPR technology to allow us to run so-called functional genomic screens that allow us to sequentially interrogate the function of each and every gene in the genome on a one-by-one basis and understand the function of those genes on the biology under study. So functional genomics takes advantage of the ability of CRISPR to specifically delete genes within human cells to ask the question, what is the relationship between the function of that gene and disease biology? There are a variety of different flavors of CRISPR, so-called traditional CRISPR, where we're using CRISPR to just simply delete the gene of interest. But there are forms of CRISPR that have now been created that allow us to either inhibit gene expression or activate gene expression. And what we're able to do in terms of functional genomic libraries is we can create libraries of CRISPRs in microtiter plate format such that each well contains a guide RNA that is targeted to a specific gene, and we can then screen those guide RNAs exactly as we've always been screening compounds to delete each and every gene in the genome on a one-by-one basis. Key to this science working are really 3 things. The first, of course, is CRISPR technology itself, which, as I say, simply did not exist a few years ago. The second is the availability of assays that we believe are predictable of disease. There's no point screening a CRISPR library or indeed any other library in an assay that isn't translatable to disease. So the creation of highly relevant disease biology assays, which we then interrogate with CRISPR to identify genes that affect the function of those cells. And then, of course, the data analysis algorithms, the imaging algorithms that allow us to extract value from those experiments. And as with much of what we do, we can only succeed through collaboration. So across AstraZeneca over the last, say, 3 years or so, we've developed a comprehensive functional genomics strategy. In 2020, we will have run around about 80 functional genomic screens. That number will increase next year to around about 120 functional genomic screens. And these apply across each of our therapeutic areas, so cardiovascular disease, metabolic disease, various lung diseases, asthma and COPD, through, of course, to oncology. And in oncology, we're running a variety of functional genomic screens, not simply to identify new targets that are accountable for cancer processes, but also to identify new targets that mediate either sensitization or resistance to our new medicines. And we do this through partnerships. So internally, we've set up a capability to run plate-based so-called arrayed CRISPR screens, and I'll touch on this on the next slide. We're collaborating with Luke Gilbert and others at the Innovative Genomics Institute to develop so-called CRISPR-A and CRISPR-I technology to study the effect of gene inhibition or gene upregulation on DNA damage repair processes in cancer. And we've established a flagship partnership with Cancer Research UK in Cambridge in the Milner Institute, where we've created the AstraZeneca CR UK Centre for Functional Genomics, which, as we move into 2021, will have the capacity to run 120 pooled CRISPR screens a year. And just to touch on the biology around these 2 different screening formats. So I've mentioned this a few times. So in arrayed CRISPR screening, we're running in arrayed CRISPR screening, we're doing very much what we've done in small molecule high throughput screening for many, many years. We're using complex automation. We're designing disease relevant cellular assays. Our CRISPR agents are arrayed in microtiter plates. So as I say, each well contains a CRISPR that will delete a specific gene or a guide RNA that will delete a specific gene. Each well contains between 4 and 6 different guide RNAs that delete that gene. And we're then able to reverse transfect those guide RNAs into cells of interest. The gene is then deleted, and then we're able to examine the effect of that on cell biology using high-content imaging assays. For pooled CRISPR screens, the paradigm is a little different. Here we use lentiviral libraries to deliver guide RNAs to cells in a pool rather than the radiant 96-well plates. And this paradigm is typically applied in oncology research to identify genes which, as I say, when the gene is deleted or the gene is upregulated, that leads to resistance to a medicine, or it leads to cells being sensitized to that medicine. And if we run a CRISPR screen, say, to identify genes that lead to resistance to a medicine like Tagrisso, which is a medicine that AstraZeneca has in the clinic for lung cancer, we may typically run a screen in as many as 10 different lung cancer cell lines. We may run that screen against different concentrations of drug and also different drugs of the same class to identify genes that, when deleted, lead to an increased sensitization to Tagrisso or indeed to resistance to Tagrisso. And the way these screens run, as I say, we screen in pools of cells. Those cells are then seeded into flasks. We look for clones of cells that grow through, and then we use next generation sequencing to identify the deletions that we've introduced into those cells. And this technology we're finding is having a huge impact on our portfolio in terms of bringing new targets into our portfolio. And as we move into next year, we hope we'll be in a position where between 15 and 20% of our new targets are identified using this methodology. And as I've said several times, critical to this is the development of the cell-based assays that we use in these screens. And to do this, we're using everything from embryonic stem cells all the way through to running screens in primary cells if we can. And this is just an example of the type of data that we get with one of these screens. So the question we asked ourselves in this assay was, which genes, when deleted, lead to loss of the androgen receptor in a prostate cancer cell line? Many of you will know that a major cause of prostate cancer is uncontrolled expression and activation of the androgen receptor, and many of the medicines in the clinic that have efficacy in prostate cancer are so-called anti-androgens. So the question we asked ourselves here is, can we identify genes which, when deleted, lead to loss of the expression of the androgen receptor in a high-content imaging assay, leading to the hypothesis if we can then find small molecules that inhibit the proteins encoded by those genes, those are potential new medicines to treat prostate cancer. So what we have on the chart here is we've tested around about 10,000 guide RNAs on this chart. The blue dots at the bottom of the chart is a control guide RNA that is specifically targeted at depleting the androgen receptor, deleting the androgen receptor. The black dots at the top of the chart are all of those genes that, when deleted, have no effect on the level of expression of the androgen receptor. And the dots that we see in between, and those that are circled in these red boxes, are genes which, when deleted, lead to a loss of expression of the androgen receptor and hence represent novel drug targets. And when we're able to identify the same genes in multiple different prostate cancer cell lines, this gives us confidence that those genes may well be directly related to the drivers of cancer in those cell lines. So in this particular experiment, we identified 54 genes of interest. Those 54 genes went through a range of further target validation studies to build confidence. And we've now started one program on one of these genes with a view to hopefully developing a clinical candidate that we will be able to take forward into prostate cancer patients. Just a couple of slides, 2 or 3 slides, just to finish off. I've spoken a lot about target identification. The challenge in all of our target identification activities is they don't give you a single gene and say, hey, this is the gene that you should prosecute. Typically, these activities give you lists of genes. The example I showed, we had 54 gene hits. It's not uncommon to get a couple of 100 gene hits. Those gene hits then need to be validated, and we move through a pipeline of parallel validation of multiple genes, the first of which is the process of in silico validation, where we're using bioinformatics methods to try and identify or to do 2 things, actually. The first is to try and identify information that may exist in the literature that would support the disease hypothesis for that gene, but also to better understand the pathways that those genes sit on. We then move through so-called in vitro validation, where we would look to probe gene function using a variety of different methods, again in cell-based assays. And typically in these assays, we're moving away from traditional 2D culture into more complex cell culture or organoid systems. And then, of course, in vivo validation using transgenic zebrafish or transgenic animals. So I've spoken a lot about target validation. In the last couple of minutes, I'll just talk about another area of huge interest for us at AstraZeneca, and this is expanding our drug modalities to target any biology. As we're all aware, the vast majority of medicines in the clinic today are small molecules or monoclonal antibodies with relatively small numbers of antibody drug conjugates, antisense oligos, and maybe a few other of these types of medicine in the clinic. And certainly 10 years ago, the development portfolios of pretty much every big pharma, every biotech, were absolutely dominated by small molecules and monoclonal antibodies. Our aspiration, the aspiration in the field, is now to make every drug target tractable. If we're going to do all of this work to identify novel drug targets, the probability is that those novel drug targets will not be tractable small molecules. They may not be tractable to monoclonal antibodies. They may sit in so-called undruggable target space. And hence, we need to be far more innovative around how we're able to drug those molecules. And that has led to an explosion in the types of drug modality that we are following in AstraZeneca and we're following in the field. Everything from the traditional small molecules and monoclonal antibodies into agents like PROTACs, which lead to the degradation of a target of interest, through a variety of oligonucleotide therapies, antisenses, modified RNAs, messenger RNAs, oligonucleotide conjugates, vaccines, of course, a variety of different protein and peptide therapeutics, all the way through to cell therapy and CRISPR. Across the AstraZeneca portfolio today, we have active programs using each of these as the proposed therapeutic modality, with probably half of the modalities represented on this slide in the clinic, and with the majority of the remainder likely to enter the clinic as we move through the next 2 years. And just to show you one example of the type of efficacy that we can see with these modalities, this is some data where we are looking to target antisense oligonucleotides to the pancreas for a number of indications, for a diabetes indication. And what we've done here is we've taken an oligonucleotide, and to facilitate uptake into the pancreas, we've conjugated it to a peptide with activity at the human GLP-1 receptor. This is a G protein coupled receptor that is expressed within pancreatic islet cells. And if you look at the data in the figure here, if we simply take the oligonucleotide in its own, we see about 50% knockdown of expression of the target gene in these cells. If we conjugate the oligonucleotide with a GLP-1 peptide, we achieve 95% knockdown of the target gene within these cells. And this data just demonstrates just an example of data on how we're able to progress some of these new molecules to the clinic, where delivery often can be a primary issue. So I will conclude there, and really just to make a couple of concluding comments. As I say, I think we live in an area of massive innovation in drug discovery. The technologies that are described on this slide, the technologies that are available to us as drug discovery scientists, allow us to generate unprecedented amounts of data and unprecedented amounts of knowledge compared to as little as 10 years ago, and really enable us as drug discovery scientists to better understand disease and better progress therapeutic models, therapeutic entities to the clinic. Key to this is understanding disease. Key to this is the ability to then create assay systems in the laboratory, many of which are based on cellular assays that allow us to identify targets and then identify molecules with activity at those targets. And that is certainly a major area of interest for us at AstraZeneca at the moment, and I think is across the field. And with that, I'd like to thank Nexelcom for giving me the opportunity to speak today, thank everybody for listening, and I think I will hand back to Lisa for the Q&A.
Thanks, Steve. That was great. Very informative presentation. So we are ready to start the Q&A portion of the webinar. If you have questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. We already have some questions that have come in. How useful are current organ on a chip models? For example, have human liver on a chip actually caught liver toxicity of compounds before they go through animal models?
So, simple answer, yes. We are using liver on a chip models routinely to do exactly that. we also are able to use a variety of hepatocyte models in 2D, which will catch the majority of hepatotoxic molecules before they move into the clinic.
Okay. This one says, great talk, Dr. Rees. Do you think antibody drugs or gene therapies will be the mainstream in pharmaceutical industry and the development of small molecule drugs will step down?
Now, that's an excellent question. So I do not believe there will be let me start again. Across the industry, we are seeing a decrease in the proportion of portfolios that are small molecule therapeutics. But I do not believe that we will see the industry moving away from small molecule therapeutics. And the reason for that is if you're able to identify a small molecule with activity at a target protein, it is relatively straightforward to develop such a molecule. It's relatively straightforward to manufacture it, and it's very straightforward to distribute it to patients, which is not true for most of the biological medicines that are currently in development. Having said that, though, I do see a significant increase actually in the number of gene and cell therapies that are moving to the clinic. I'm especially excited by the potential of CRISPR as a medicine. The first CRISPR medicines are now in clinical studies for diseases such as beta thalassemia and sickle cell disease. Those medicines are currently ex vivo gene therapies, and in those first clinical studies, the initial patients were effectively cured of disease. And I'm hugely excited by what CRISPR potentially can do as a medical entity in times to come, maybe more so than traditional gene therapies. And also in terms of cell therapy, and particularly oncology cell therapy, I think we are very much at the cusp of what's being achieved with so-called CAR-T therapies. Most of those therapies today are so-called autologous cell therapies, where cells are taken from the patient, they're modified and they're reinfused into the patient. With the development of stem cell technologies, with the development of CRISPR to then modify those cells such that they're, in essence, to increase efficacy in the patient population, and maybe to generate cell therapies that are so-called, sorry, I forgot the word, but cell therapies that we can dose to every patient. I think we will see a huge explosion of that technology as we move through the next 5 to 10 years.
Okay. The next question is for Steve again. Has AstraZeneca been doing work to create off-the-shelf cell therapies, for example, allogeneic CAR-T therapies? And if so, have current preservation techniques posed challenges?
That's a good question. That picks up on what I've just been saying. So the ability to create allogeneic cell therapies for cancer is something that we have a huge interest in, but that's very much work that is just initiating at the moment. Regarding the preservation, has that caused challenges? I don't think I'm the best person to answer that question, but conceptually, I don't believe it should be a challenge.
Okay. This person says, I know that the focus is on the body itself. How can all these gene techniques be applied to fight against the silent pandemic of antibiotic resistance?
Antibiotic resistance. So that's another very good question. So how about antibiotic resistance? So as a company, not surprisingly, we do very little anti-infective work, or at least we did very little anti-infective work pre-COVID. But the basic technologies, I think, So this morning, coincidentally this morning, I'm a member of the Scientific Advisory Board for the anti-infective group at the University of Dundee, and we had our annual SAB meeting this morning. They are using exactly the same technologies. They're using functional genomic screens, they're using CRISPR, they're using multiomics, they're using chemical biology methods to identify new drug targets in diseases such as Chagas disease, trypanosomiasis, and the like. So I think the high-level answer to the question is that these technologies are equally applicable to the ability to discover new antibiotics, to discover new medicines for neglected tropical diseases. I think the challenge in many of those diseases is maybe building that expertise within those organizations that are developing medicines against those targets, such that we can see those benefits.
So we're getting a lot of really big picture questions here.
They are quite big picture questions, yeah.
Yeah. Do you think it will be possible to have advanced enough cellular models to test the efficacy of drugs that you won't need to test drugs on animals at all?
So that's another very good question. And yes, I do. Well, yes, I believe that it will be possible. Well, actually, I believe it's possible today to create cell-based assays that allow you to test the efficacy of drugs without going into animals. And I believe we do that routinely for our cancer medicines. However, it is a requirement today that all medicines are tested in animal models for toxicity, and that I don't see changing for quite some time.
Okay, thanks.
And that's a legal requirement.
Yeah. Do you think that monoclonal antibodies represent the medicine of the near future?
I think monoclonal antibodies represent the medicine of the now. If you look at the top 10 selling medicines globally today, the majority, 6 or 7 of them, are monoclonal antibody medicines. I think the challenge in the monoclonal antibody field is the development, like everything. Many of the monoclonal antibodies in the clinic today were identified against very well-validated targets, and also targets that were directly amenable to that approach. I think the challenge in the antibody field today is the same as the challenge in the small molecule field, how we then better validate our targets from monoclonal antibody approach. And maybe many of these targets, like integral membrane proteins, are that bit more challenging for the successful generation of a monoclonal antibody therapeutic.
Okay. Next question. CRISPR screen is usually done on cell lines, and cell lines are rarely representative of the tumor. How do you go around? Multiply cell lines or attempt screens on primary samples, or validate out of a large set of hits?
Yeah, so today the answer to the question is multiple cell lines derived from the same tumor, and look for common genes that are common to that particular tumor type. We and many others in the field are beginning to run CRISPR screens in primary cell lines. Others are beginning to look in tumors. There's also a number of groups that are beginning to run whole animal CRISPR screens. We are not yet convinced that we would need to go into more complex systems, but we're running a number of exploratory experiments to explore whether we need to do that.
Okay. Another CRISPR question. Do CRISPR screens help identify specific patient populations where a drug will or will not work? For example, do they inform treatment recommendations?
Oh, the latter part of the question is the answer no today. I'm not aware of CRISPR screens directly informing a treatment recommendation. What CRISPR screens are able to do, as an example of what CRISPR screens can do, we are running CRISPR screens to identify the likes of the clinical mutations that are likely to arise to our medicines. So, as an example, AstraZeneca launched a medicine a few years ago called Tagrisso, which specifically inhibits the tyrosine 790 methionine mutation in the epidermal growth factor receptor. The primary resistance mechanism to Tagrisso is a mutation of cysteine 967, I think it is. I might have the number wrong, but a cysteine within the epidermal growth factor receptor. We're able in the laboratory to predict that is the mutation that would arise following Tagrisso treatment by treating cells with Tagrisso. We're now doing that routinely such that we can predict the clinical mutations that will arise to our medicines, such that we can then begin to consider what future medicine we may need to generate that would address that clinical mutation. That's not quite the same experiment as is described or as was posed in the question, but I don't think. I'm just reading the question again. Identify specific patient populations where a drug will or will not work. No, I don't think CRISPR will help there. That is where obviously next generation sequencing and the ability to stratify tumors at the genetic level has had a massive impact and is impacting today.
Okay. The next question is, do you include scRNA-seq in your genomics area of development? It says many bulk signatures are co-founded by averaging.
Sorry, let me just find that question and read that question.
Question number 3.
No, yeah, I found it. So no is the answer today. We are initiating, so single-cell RNA sequencing is a technology platform that I think is very much in its infancy. It's advancing very rapidly. I'd say for us in AstraZeneca today, single-cell sequencing is a technology that we are exploring rather than being integrated into any specific project. But I absolutely take the point of the questioner. We do from our own data, and it's obviously common in the literature. We see very significant differences across different cell types within a tissue when we move into single-cell RNA sequencing.
Okay, so the next question is also another big picture question. It was asked early on while you were presenting, and it is, what is the next revolutionary technology that you believe will turn science into medicines?
Now, that's a very good question. This is a can you predict the future question. What is the next revolutionary technology? So something that we are incredibly excited about, and also others in the field are incredibly excited about, is the application of artificial intelligence and machine learning in chemistry. And this is probably the area that excites me most. So using artificial intelligence in chemistry, we're asked two questions. First question is, can we use AI to tell us which molecule to make next? And then can we use AI to tell us the best route of synthesis to make that molecule? And we know that the answers to both of those questions are yes. What we don't know is whether we can do that to dramatically shorten the timelines from the identification of initial hit through to the delivery of a drug candidate. Our prediction is over the next 5 years that we will be able to reduce the timelines from hit identification to candidate selection from the current 3 to 4 years to maybe less than 12 months through applying AI in chemistry. And we're also looking at applying similar AI in our antibody optimization. It's not just simply unique to small molecule discovery, but this technology will hopefully dramatically decrease cycle time in terms of our ability to get medicines to patients quickly, but also increase success through allowing us to identify molecules, deliver clinical candidates for those projects that might have failed.
Okay. Wow, lots of great questions
That's
today.
Lots of great questions. That's probably the best set of, I congratulate everybody on the call. That's probably the best set of questions I've ever been asked in one of these presentations.
Yeah, a lot of big thinkers out there today for this talk. So I would like to thank you, Steve and Jean, for your presentations. And I would like to thank everybody for joining us today for this presentation. We ask that you complete a short survey at the close of this webinar. Your thoughts and comments would be greatly appreciated, especially from such big thinkers that we have on here. Make sure you visit the poster hall and the exhibit booths where you can learn more about a variety of exciting technologies that we have. And this webcast can be viewed on demand through July of 2021. Until next time, thank you very much and have a great day.